Okay. Okay, so good morning, good evening, everyone. So welcome to today's ImmuZoom seminar. So it's really my great pleasure to welcome today's speaker, also my mentor, Dr. Wanjun Chen from NIDCR, NIH. So before the seminar, I just want to give a brief introduction of Wanjun. So Wanjun is now a senior investigator, also chief of mucosal immunology section in NIDCR, which is National Institution of Dental and Craniofacial Research, NIH. So Wanjun received his medical degree in China and did his postdoc fellowship in USA Harvard Medical School. His research mainly focused on the machinery of T-cell immunity and tolerance and manipulating T-cell immunity versus tolerance in animal models in order to understand the pathogenesis and the development to develop immunotherapy for autoimmunity, cancer, and infection disease. I think most of us actually are very familiar with Wanjun's long history of research and also his career, particular with his tremendous contribution in TGF-beta biology. Among his very many scientific contributions to biomedical research, Wanjun is the very first one who actually did discover that TGF-beta can induce 4p3 in naive T cells and then convert them into regulatory T cells. I think this is very much the highlight of his whole and the center of his whole research program. And this very original paper describing the TGF-beta inducing t-reg cells was originally published in JAXMed, and then currently actually being cited over 55,200 times, which is, I think, very remarkable. I mean, obviously, besides this this main discovery, Wanjun also has a lot of very breaking through findings, and then he published over 120 articles in all the international journals, including Nature, Nature Medicine, Nature Immunology, etc. So among these achievements, Wanjun has received many honors and awards, including Scientific Achievement Awards from NIH Asian and Pacific Island American Organization and Wang Yinline Memorial Lecture Award. In addition, he also was selected as president of Society of Chinese Biologists at USA. Washington, D.C. Baltimore chapter in 2015, and now serve on the Council for the National Society of Chinese Biology at America. So he has been elected as member for the membership and the Communications Committee, International Cytokine and Interferon Society for 2021 to 2023. I think without further ado, I think we are very much looking forward to your lecture. Please go ahead.
Thank you, Chen, for the kind of introduction. I think somebody called iPhone recent hand. Chen, you might want to, I don't know what he wants to do or she wants to do there. Thank you for the kind introduction. What I'm going to do today is try to describe or talk about some work in my laboratory during almost 2 decades about the development and function of t-regs or T-regulatory cells. So I'll start with this brief introduction of these so-called regulatory T cells, or T-regs for short. We know that there are several types of regulatory T cells have been identified by today, but today I'm only focused on this particular one, which is the CD4 positive, 25 positive, and CD4 positive T-regs. We know now that these T-regs exist in both human and mice. They play an essential role in immune tolerance because without T-regs, such as the FOX3 gene knockout mice, you can see here, created by Sashudinsky. These mice will develop massive and uncontrolled inflammation. All the mice will die by a few weeks of age. In addition to that, we also know T-regs are important in terms of the inhibiting or suppressing anti-tumor immunity. Almost all the patients with the cancer have increased the frequency or number of the T-regs in the body, especially in some tumors in the intratumor tissues. That's quite remarkable. Another reason I want to give you an example. We know that the cancer, so-called cancer immunotherapy, is one of the most exciting area, not only by academic, maybe also by pharmaceutical company. We also know several so-called immune checkpoint blocking antibodies have been approved by FDA. Also, I think by China's FDA too. And these antibodies have been used for treatment for the different cancers. Well, I give you one example, which is CTLA-4 antibody. We know this antibody is the first antibody approved by FDA. We know this antibody also effective in treating certain cancers, especially like melanoma cancer and lung cancer. However, what is the mechanism by which this CTLA-4 antibody treats the cancer by immunotherapy is still remain largely unknown. At the beginning, people thought this CTLA-4 antibody is to block the CTLA-4 on T cells, therefore enhancing anti-tumor immunity. But later on, people found prove that's not the case, because one of the important reasons for this CTLA-4 to work in the anti-tumor immunity is to selectively deplete or reduce the intratumor T-regs. Although now, what is the mechanism by CTLA-4 antibody to deplete or reduce T-regs still remain largely unknown. But in this one, we can at least see T-reg study still is an important area. So my laboratory actually, in this particular project, we focused on three aspects. One is the development and generation of T-regs. Second is the suppressive mechanism by which T-regs suppress immune response. A third one, recently, we want to manipulate T-regs for developing immunotherapy for autoimmune diseases and cancer. So I'm going to try to highlight some of the findings that we had so far. Depending on the time, I do not want to spend too much. If time is not allowing me to do anything, I can stop at any time. First one is, as Tran mentioned, that in 2001, which is 20 years ago, we discovered actually for the first time the TGF-beta can induce so-called anergic suppressive CD4 positive, 25 positive, CTLA-4 positive T cells. I want to remind the young people, this is the name of the T-regs before the T-regs was coined later on. So I gave a talk in Hawaii that year about this work. The abstract described how we discovered TGF-beta can induce naive T cells from naive T cells to become T-regs, even though we did not know the molecular mechanism, how that happens. Now, very fortunately, actually, immediately followed the discovery by the three independent groups, showing that the FOX3 is the specific gene and the transcript factor for the generation of T-regs. During our revision of the paper, we immediately realized that maybe the TGF-beta induced the so-called anergic suppressed T cells is kind of T-regs inducing by induction of the FOX3. That was the case. This is the original data you can see here. If we take the CD4 positive naive T cells, and sorted by machine. Then, though, although the machine was very slow, I remember one day we can sort 1 million cells, and we can stimulate these cells by TCR stimulation at the different doses of TGF-beta. You can see very nice FOX3 induction by TGF-beta. I have to say at the beginning, we were surprised, everybody was surprised. Most people did not believe. Now, as Chen mentioned, that this paper now is cited by many people and is recognized by all the laboratories in the world. I'm also happy to report to you that this paper has been listed as one of the 5 most highly cited papers in JEM during the 125-year anniversary. You can find that that information on their webpage. So because of time, I only use this to summarize briefly. We discovered that TGF-beta as the essential factor in the context of TCR stimulation induces FOX3 from naive CD4 positive T cells and convert these cells to become FOX3 positive Tregs. This happens in vitro and also in vivo. I may come back to this point later on. You can call that I-Tregs, P-Tregs, whenever you want to call. Despite the general consensus that TGF-beta is absolutely required for the peripheral Treg generation, the function and the role of TGF-beta in the development of the so-called thymic Tregs or natural Tregs remained somehow in debate, at least for a while. Therefore, we decided to address this question, and step by step, we now believe TGF-beta also play an essential role for the t-reg generation. I use the following 10 minutes or so to highlight how we came to this conclusion. As most people know now, that the generation or development of the Tregs in the thymus has been an important topic. There were several of the models or theories try to explain how that t-regs are generated. One of them, which is dominant one for a while, is that people believe that thymic Tregs are developed by so-called high-affinity TCR driven development of Tregs, or so-called agonist selection of Treg cells in the thymus, meaning somehow some T cells, when they see their high-affinity TCR in this particularly high-affinity area, instead of committed negative selection, these somehow become Tregs. This is a very important model, and it explained a lot of things, and also supported by many evidence. However, there are several puzzles, and this model cannot explain. For example, later on, several studies show that not only the high-affinity TCR, low or medium affinity TCR can also generate Tregs in the thymus. Second, if high-affinity TCR is the intrinsic fit determining factor, why do only some thymocytes become Tregs, others die? Does negative selection have any relationship to Treg generation? If so, what is the missing link? Third one, this model cannot explain, which is a very intriguing one. We know in the normal mice, we cannot detect the Tregs until day 3 after the mice are born. This is a very strange day. What happened by this day 3? That's the question this model cannot explain. Keep these questions in mind. We asked whether TGF-beta plays any role in the t-reg generation. For that purpose, we made this so-called TGF-beta receptor 1 T-cell specific enough mice. In these mice, the T-cells lost their TGF-beta receptor 1, therefore cannot respond to TGF-beta, and the other cells are still intact. And these mice, believe it or not, is very striking. And almost all of them, and I think all of them, develop lethal inflammation, die by 5 to 7 weeks of age. In the periphery, there's almost no Tregs there, suggesting the TGF-beta signaling T cells is quite striking. It's important to keep this mice alive of the mice. The important question is what happened in terms of the t-regs in these thymus. What we did was we took this D3 or 5 age of the older mice and isolated their CD4 positive, CD8 negative single positive thymocytes. We know this is where we can find the Tregs. As reported by others, in the white type mice, you can see very nice population Tregs are now coming. However, without TGF signaling in T cells, these cell Tregs cannot, almost nothing there. This data suggesting TGF-beta play an important role for the normal Treg generation, especially at the neonatal age. Some people may argue that we know the LCK query or CD4 query is not a perfect query, maybe have some kind of leaking. So we continue to confirm this question by using another approach. We use this so-called mixed bone marrow chimeric mice. What we did was we deleted the TGF receptor 2 or receptor 1 in bone marrow cells, use the ER query, and mixed with the normal wild type bone marrow cells, and inject these bone marrow cells into a sublethally irradiated ROC1 knockout mice. Wait for 5 weeks. This is something relative to the neonatal age of the normal mice, and then isolate the single positive thymocytes and check what happened in Tregs. As you can see here, while the white type thymocytes show very nice population Tregs already there, but there's almost nothing in terms of Tregs in this TGF receptor 2 knockout mice. And if we put the 3 mice together, that's what we found. Almost nothing, this both 25 negative or 25 positive, FOXP positive Tregs. This data together confirm that TGF must play an important role in the t-reg generation. Based on that data though, we proposed by then, this is a long time ago, that the TGF-beta in the context of TCR stimulation in the thymus, especially the t-reg precursors, most likely the single positive thymocytes, can induce FOXP3. Once these FOXP3 are induced, it is R2 in the thymus to maintain or keep them alive in a very restricted, balanced manner, because we know that in the normal thymus, we cannot find very much R2 there. And after this, the 2008 paper was published, another group independently confirmed the role of TGF-beta in the development of Tregs. However, they had proposed a different mechanism. They proposed that TGF-beta instead play a role in the induction of the FOXP3 gene. They proposed that TGF-beta may selectively or specifically protect t-reg from death, which is quite a nice proposal. Now, however, many years ago, I published a paper actually, 2001. We showed that TGF-beta can protect the T cell from death, including the thymocytes. However, whether TGF-beta can selectively or specifically protect the t-reg from death, not the other population cells, is the question. To answer that question, we made these mice. This is a FOXP3 query TGF receptor 1 mice. In these mice, the TGF receptor 1 was intact until FOXP3 is induced. The hypothesis was, if TGF-beta specifically protected this Treg from death, we would like to see these knockout mice should have a profound decrease of Tregs in the neonatal or adult age. However, when we look at these mice though, no matter in the neonatal age or adult age, there's no difference at all in terms of this t-reg in the thymus, suggesting this is not the case. In limiting that TGF-beta can be selectively or specifically protect the t-reg from death. Now, with that data though, we had to come back our original idea, which is the TGF-beta may induce FOXP3 in the t-reg precursor in the thymus. To answer that question, to test this hypothesis, we used quite different approaches, which is done by a very talented postdoc, Joanne Cockross, who now was back to the UK, became a professor herself. And she used quite different in vitro, in vivo, physiological, non-physiological approaches to prove this is the case. Because of time, I show you two examples. One is the intrathymic injection for this t-reg precursor in thymus, in the normal thymus. What we did was, we took the CD4 positive, 25 negative single positive thymocytes from DU11 RAG1 knockout mice. We know in these mice, there's no endogenous T-regs there. We inject these cells intrathymically with their specific peptide, which is the oral peptide, in the absence and the presence of anti-TGF-beta antibody to neutralize TGF-beta, wait for 18 hours before the cells can proliferate. We took these injected transgenic T cells in vitro to test the FOXP mRNA to see what happened. As expected, without injection, of course, there's no FOX3 mRNA. With this injection, you can see here, very nice FOX3 mRNA induced there. This is the transgenic T cells. This induction of FOX3 was indeed because of TGF-beta. Because if we neutralize TGF-beta in thymus, this induction was almost completely abrogated, suggesting TGF-beta indeed play an important role for induction of this FOX3. Now, some people may say, okay, this is transgenic T cells, may not reflect the real non-transgenic mice. Now, to answer that question, we did the following experiment. What we did was, we took the CD4 Cre TGF-beta receptor 1 double negative, either control or knockout thymocytes, together with this mix them together in the 1-to-1 ratio. We know that double negative cells in the CD4 Cre, these cells has not been yet deleted their receptor 1. Intrathymically directly inject into the host of which is the normal black cyst mice, waited for 12 days. This is what we can find the normal single positive thymocytes developed. And then we took the single positive, CD4 positive thymocytes, look what happened in terms of these regulatory T cells. As you can see here, Y-type mice, Y-type, the T-regs derived from the control double negative thymocytes show very nice population T-regs there. However, without TGF signaling, you almost cannot find any T-regs there. This is the frequency. This is the total number after 12. You can calculate that. This together strongly suggests TGF signaling indeed is absolutely required for the FOXP gene expression and the T-reg generation thymus. Now, the next question is, now if you think TGF is important, then where and how is TGF produced or increased in the thymus? To answer that question, we did first a very simple experiment. We took very normal mice, their thymus, and get their protein, did the very simple TGF-beta1 or 2 of the ELISA to measure the TGF protein. Either this is the fetals or the day 1, day 2, day 3 neonatal mice. As you can see here, even if the fetal mice, they have a TGF-beta in the thymus. However, with age, especially by 2 or 3 days, you can see the total TGF-beta protein is increased in the thymus, in the normal mice. But we know that the total TGF-beta protein does not necessarily reflect the activity of TGF-beta because TGF-beta needed to be activated in order for TGF-beta to signal. Therefore, we did the following experiment. We directly use the immunohistology staining to detect the in situ, the active TGF-beta by this specific antibody, which kindly provided by Dr. Flanders from NCI. This antibody is not commercially available unfortunately, but this is a very unique, very effective antibody. As you can see here, in the fetus or day 1, day 2, or day 3 older mice, this is normal mice thymus. You see here, even for the fetals, you see some spotting there and positive TGF-beta positive cells, but not many. With age, especially by 3, you can see the active TGF-beta was dramatically increased in the thymus. Another interesting point is this TGF-beta staining is not universally distributed everywhere. They are localized in certain area, but if you look at more carefully, these areas are mainly the medulla area or the edge of the between the cortex and medulla area. We know this is where we can find the T-regs there, suggesting that indeed for some reason, by 2 or 3 days, this TGF-beta can be increased and also the active TGF-beta is increased. With that data though, I'm sure the people will immediately ask, can you tell me how that TGF-beta is increased? Now, we found this paper. I read this paper which was published in 1994 by Charlie Su and Jenderson Sprint. I'm sure some of you know that unfortunately, very talented Charlie, who is also my good friend there from South Korea, he died by cancer a few years ago. Very talented. In this paper, the elegantly show the normal process of apoptosis in the thymus to resolve some paradox. Normally we know that more than 90% of thymocytes become dead. However, at any time point, if you cut the thymus, do a staining, you cannot find very many apoptotic cells in there in the thymus, because this is the paradox for many years. But this paper, they elegantly demonstrated what happened there. I quote, this is their original statement. He said, thinner staining of the fetal thymus is quite low. By day 2 after birth, however, staining is as prominent as in the adult mice. By day 2, very magic date. And also in this paper, they showed very elegantly apoptotic thymocytes can be read very rapidly and sufficiently engulfed and digested by thymic macrophages. Different macrophages from cortex to the medulla, but this resolved why at any time we cannot find many apoptotic cells, because they are all gone. Now, with that paper, we hypothesized and also proved in this paper, because of time I highlight here, we showed that apoptotic thymocytes could trigger thymic macrophages, dendritic cells, even epithelial cells to produce TGF-beta. I showed you already TGF-beta can induce in vivo in vitro FOX3 in this single positive thymocytes. That's what we showed there. And if that's the case, suggesting apoptosis can somehow have a relationship to the generation of t-regs there. Now the question then was, if that's so, can we change the apoptotic level? Therefore, we can change the t-reg generation of the thymus. To answer that question, actually, we did quite a different experiment. I'll show you a few. 2 sets. One is the adult mice, another is neonatal mice. I thought this experiment is very talented. Postdoc discovered this talent, set up this experiment. I think this is a very smart one. What she did was she took DU11 t-reg1 bone marrow mixed with the normal BARBC bone marrow, which is 25/75, and they inject these bone marrow cells to sublethally irradiated DU11 t-reg1 mice, and then wait, and then immediately the second day inject either PBS alone or PBS alone or 200 milligram of over protein. The purpose for that one is to induce apoptosis of these DU11 thymocytes, and then wait for 3 weeks, check what happened in the B-black-cyst T-regs. The hypothesis was this over protein, which is 2 milligram, can cause apoptotic cells in these DU11 thymocytes. These apoptotic cells could trigger more BARBC t-reg generation in the thymus. That's exactly the case. As you see here, in the PBS injected, you get a good, nice population. However, with over-injection, which have a high level of apoptotic cells, you can see here, there's a significant increase of apoptotic cells. Therefore, increased apoptotic cells in the thymus could induce more Treg generation. Conversely, on the other hand, though, we also show that decrease of apoptotic cells could decrease Treg in the neonatal mice. We have done several experiments. For example, in vitro, we can use the F-toll by inhibiting the apoptotic cells with the caspase inhibitor. We can find that Treg generated was a generation of Tregs was decreased. In the normal black mice, you can do that yourself. Inhibiting this apoptosis by injecting this inhibitor, you can see the Tregs are reduced too. Also, we took this beam knockout mice in the neonatal age. We know beam is the important apoptotic pathway. Without beam, the thymocytes cannot get similar level of apoptosis as wild type mice. At the neonatal age, either D3, D5, we demonstrate that they have less apoptotic cells, they have less TGF production. In the paper, you can find that. And also the Treg cells was reduced too, either total number or percentage. I have to remind everybody, when these mice become adults though, the Tregs are higher in the thymus because these people knockout mice, the Tregs are less likely to become apoptotic. This is together make us to say that the changes of thymic apoptosis indeed can affect the Treg generation. Now, the final question now is that if that's the case, now what is really the role of the affinity of TCR or strength of TCR in the Treg generation? It's really that the affinity of TCR instructs the precursor of Treg cells to become Tregs, or something else. Basically, the question is, this is the qualitative or quantitative? To answer that question, we use this approach again. This is intrathymic injection directly. We took this CD4 positive, 25 negative, zinc positive thymocytes, which is the 567 ROG1 knockout mice, and injected one macro peptide together with these cells. This peptide contains either high affinity K5 or intermediate PMMC or low affinity 102S peptide. These three peptides all can be recognized by these transient T cells into beta-A thymus and see what happened in the Treg generation. As you can see here, no matter it's high or low affinity of TCR, they all generate Tregs. The difference is the amount of Tregs generated. As you can see here, the high affinity of the Tregs peptide indeed generate more Tregs than the low affinity, but everybody can generate Tregs, meaning that high or low affinity is the factor, but not the intrinsic factor. Importantly, no matter it's high affinity or low affinity, TGF-beta plays an important role. If we co-inject intrathymic with anti-TGF-beta antibody, you see the generation of Tregs was significantly reduced. With that together, we propose the following model, try to explain how that the t-reg generated in the thymus. We believe apoptosis in thymus through the TGF production can drive FOX3 expression, namely that the negative selection may trigger the t-reg development. Now, this model, I spent 1 or 2 minutes to go through with you, which is, I think, is important. We believe apoptosis either by the high affinity TCR stimulus, which is the negative selection, for example, or by other apoptotic stimuli. These apoptotic cells can be digested or trigger thymic macrophages, dendritic cells, even epithelial cells, produce a transient gradients of TGF-beta in rich environment. Remember, this is the transient gradients of TGF-beta in rich environment. Now, once this environment was established, for example, if some precursors, as here, this cell is in the right location with a sufficient amount of TGF-beta at this time point, this is very important, can see there's passive peptide. This can be either high, medium, or low affinity TCR, but not too high, become negative selection, not too low to be ignored. these t-regs can stimulated by TCR and TGF-beta to become t-regs. Now, another scenario is this same precursor. If at this time, they can also see their TCR. However, if this cell is outside of this TGF-beta milieu, which means no TGF-beta presence, I do not think this cell can become a T-regs. They can do whatever they needed to do, either become normal thymocytes or become dead, depending on their TCR strength. The third scenario is if another precursor, which is in the right place with sufficient level of TGF-beta, but at that time, if this precursor did not see their TCR, this cell will not become T-reg. They will stay there whenever they want to. Now, can this model is to explain several of the models. For example, we know that once the T-reg are generated, our Syngus group very nicely shown that the FOX3 is intrinsically to promote the T-reg from death until you have a sufficient level of two is there, meaning this one can control the level of T-regs in the thymus. Now, this model together, though, I think we can very well reconcile with so-called high affinity TCR mediated T-reg generation. Rather than thinking that the high affinity TCR intrinsically trigger the T-reg generation from the T precursors, we think this high affinity TCR may be generating more T-regs by induced more apoptotic cells thymus. The second is, this model can also very nicely reconcile with so-called delayed appearance of T-regs in the normal thymus. I said earlier today that the mice D3 only D3 we can detect the T-regs, but not earlier D1 or D2, because as Jonathan Spring in their paper, for some reason when the mice are born by D2, the apoptotic level in the thymus suddenly become very high. We do not know what happened. These apoptosis cells increase the level of TGF-beta may get more T-reg generation. This model can also very nicely reconcile with so-called intraclonal competition for T-regs, because we know in this model, people believe more precursor for T-regs, less T-regs per se, the percentage were generated, because these precursors, the more T-reg precursors can compete the limited amount of TCR. We think not only compete for limited amount of TCR, maybe they can also compete for the limited amount of TGF-beta, because as I said earlier, this TGF-beta is tracing is not permanent there. However, this model reconcile with so-called two-step model of T-reg generation by R2. We are currently addressing this question, but I can tell you this model, their findings very well, perfectly correct. However, their interpretation of the data though has some kind of inaccuracy. We believe that one can very well with the oral model there. This is still ongoing work. If possible in the future, I can explain to you later on. Therefore, I would like to use this commentary I wrote a few years ago, made this statement. I think now it might be the time for us to reconsider the possibility that T-reg cells are all generated with similar pathways. The major difference between the so-called T-regs and the P-regs would therefore be the location in which the T-regs is generated, and as such, the antigen drive their development rather than the requirement of TGF signaling in their generation. This is my statement. Whether this is true or not, we need to wait for more time to be confirmed by other group, but we are confident this is the case. So since I used already 30 minutes or so, I will skip this suppress part and come to my last, which is the I would share with you some kind of the effort we have done recently by manipulating the T-regs for immunotherapy treatment. Basically, most of them is autoimmune diseases. Now, if you ask me, can T-regs be used for the immunotherapy in autoimmune diseases, my answer is yes. No question about that. Someday we will do that. We can do that. However, if you look at the literature, the approaches people have used so far is possible to develop this kind of immunotherapy. It's very difficult to judge. For example, can we just simply isolate the T-regs from the mice or from the patient in the future to adopt transfer back to this patient to treat the autoimmune disease? My answer is no, for two reasons. One, we know that T-regs, whenever you isolate from the normal mice or human, is the pool of T-regs recognize different type of the antigens. This contain some T-regs suppressing inflammation, also contain T-regs suppressing anti-tumor immunity or anti-cancer immunity. Therefore, it's not a very good idea to just go back to expand these cells on specifically and adopt transfer back to the patient. And the second important point we have to consider is patients. It's not like normal people or not like naive mice. In this patient, especially the autoimmune diseases patient, they have something wrong in their body. They have inflammation around their body. We know now T-regs can be changed to another phenotype or phenotypes during the different environment which contains inflammation. For example, if IL-6 may change the T-regs become Th17 cells. The second possibility is now in that case, can we adopt transfer some in vitro generated antigen specific T-regs, which is possible. However, we have to resolve two important questions. One is that we have to make sure this in vitro generated T-regs, for example, by TGF-beta with TCR, they are stable in vivo. Second is, as I mentioned, we have to be sure the recipient do not have much of the inflammation in their body. The third possibility is, can we in vivo generate antigen specific T-regs by, for example, give a low dose of peptide? Because there are several groups beautifully published several paper showing that this happens and worked in the naive mice, but not in the mice with established diseases. Therefore, it's not possible for the patients with diseases. Another possibility is now in vivo expansion of T-regs, for such as by IL-2. I think this is possible because there are several clinical trials going on. There are some promising results. However, we have to be cautious because this approach is based on the fact that IL-2 can bind the 25, which we know in the normal situation, most of the 25 positive cells are T-regs. Therefore, they can selectively expand T-regs. However, we have to keep in mind, this IL-2 does not recognize which T-reg is against the inflammation, which T-regs is against the anti-tumor immunity. Therefore, there's still something we needed to be more careful in the future. Now, I'm sure some of you know that there's the CAR-T. I personally also like this idea, but in order for us to do anything in the future, we have to be sure. Currently, maybe the self CAR-T may be possible, but the allogenic CAR-T, no, we have to cross that barrier, whether this CAR-T T-regs can work on the survive in the recipients. Therefore, the real challenge we are facing is how to generate these autoantigen specific T-regs in animals and ultimately in patients with established autoimmune disease. This is very important, okay? When the patient see their physicians, they already something wrong in the body, not the normal people anymore. To specifically suppress the inflammation in the organs, tissues affected, and importantly, without compromise their overall immunity and also the especially anti-infection and anti-cancer immunity. That's the challenge and also ultimately the goal for us to reach. So Before I show you some data, I would like to remind you that we and others and show by many experiments, both in vitro and in vivo, with the apoptotic cells. But remember, this is apoptotic, not necrotic, or not the, we do not know about apoptosis. One time I talked to Xiaofeng with this question. Actually, we do not have any evidence yet, but for sure, apoptotic cells can trigger macrophage or immature cells produce TGF-beta or Thymic PGA2 without producing pro-inflammatory cytokines such as TNF-alpha, R1 beta, or R6. In contrast, we do the opposite. Therefore, we would like to see whether we could use this mechanism to develop immunotherapy by inducing antigen-specific Tregs in vivo, together with the fact that TGF-beta is the key factor to convert naive T cells to become Tregs. With this question, actually, we developed this approach. Now, the underlying mechanism of how that works was this, okay? We know if we look at the autoimmune diseases, most of the organ-specific autoimmune diseases, except for lupus, despite this autoimmune disease, they have different symptoms, different pathogenesis, different organs affected. But if we look more carefully at the bottom of these diseases, we would like to say there's a common underlying mechanism, which is this patient have a dysregulated immune response. The balance between immunity and tolerance was broken. In this case, if it is very difficult, if not impossible, to really develop some kind of effective, even curative approach to cure the diseases without using some kind of radical approach. Therefore, we developed this idea. We thought, okay, if that's already dysregulated, can we first break down this dysregulated immune system and then try to reprogram it? Now, with this idea, depending on the two facts I just mentioned to you, TGF-beta and phagocytosis, we developed this approach, which can be artificially divided into three steps. Once tresent induce large number of the immune cells become apoptotic. That's tresent in vivo. For two purposes. One is empty some space for the immune system, let the new cells to have a space to live. Second is provide a good meal for the phagocytes. Once the phagocytes, especially macrophages, take up and eat these apoptotic cells, they can produce a tresent but a large amount of TGF in vivo. Now, once this TGF is produced, this can create this tresent immune regulatory milieu. Once this milieu is established, we could administer that specific peptide whenever we needed to. For example, EAE, we can choose the MOG. Type 1, we can choose the GAD65, so on and so forth, to trigger these naive T cells to direct directly to these antigen-specific Tregs, but not the infected cells. This is, sounds very complex. Actually, it's very simple. Three key factors. You have enough sufficient tresent apoptotic cells, phagocytes presence, and also specific peptide. Now, this is the, I have to be honest, at the beginning, we did not expect this can get a very significant effect, but this result was very surprising. This is 2014, we published this paper in Science Transgenic Medicine. We showed that in vivo, we can generate antigen-specific Tregs and treat autoimmune disease without compromising antibacterial immune response. In this paper, we show that using apoptosis, either CD4, CD8 T cells, or B cells with different peptide, we can achieve long-term suppression of autoimmune disease, such as EAE and type 1 diabetes. We have evidence to prove that autoantigen specific Treg cells are generated in vivo, and the therapy, importantly, does not compromise this antibacterial immune response. So, following that study, we continue to use this approach, modified approach. Actually, this is the animal models we tested so far. And we all get very striking and nice results. This disease can be very efficiently suppressed. This is the publication related to that. Because of time, I'm not going to through. I can only show you one example, which is the EAU, which is the uveitis in the eye, autoimmune disease in the eye. This is a close collaboration with Wanjun Chen from Zhongshan University. Unfortunately, Wanjun Chen passed away a few months ago. I'm sure some of you knew that tragic death by her, which is great loss for us as immunologists. What we did was that we immunized these mice to induce EAU. If 14 with the disease occurs, we give one time 100 micrograms anti-CD4 antibody injection, depleted T cells, and then give these mice 6 times low dose RRBP specific peptide, which what's going on in these diseases. This is the very typical experiment, as you can see here. Without treatment, these mice get very severe diseases. If only give CD4 antibody, deplete T cells, but with irrelevant control peptide, you did not get very good suppression. Although at the beginning, some suppression was occurring there, but ultimately these diseases come back. However, if you use the CD4 antibody together with specific peptide, you get a very nice suppression of these diseases. If you look at the eyes, for example, day 28, you took the eyes and look at what happened in terms of the cytokines. You can see here, for example, the TH17 cells was significantly reduced, although at this time, interferon gamma did not change very much. And inside of the eyes, you can see the FOXP positive T cells was significantly increased. Although we cannot see this antigen specific, but in the paper, we have the evidence to show this approach indeed induce the this antigen specific T-reg there. And another important question we want to ask is, if that's the case, can we, for example, to use this approach to suppress autoimmune diseases, but without compromising this anti-tumor immunity, because we did not have evidence before. For that purpose, we did another experiment, similar approach. As you can see here, well, 1 month later, the disease was very nice suppressed. At that time, though, we inject these 3 groups of mice with melanoma and wait for another month, see what's going on. As you can see here, at the day almost 2 months later, the disease was still suppressed in this specific treatment group. And importantly, we look at the development of the tumor in these 3 groups. There's no difference at all. Therefore, this treatment suppressed autoimmune diseases without compromising anti-tumor immunity. I think I already spent like 40 minutes or so. I have one story there. But
it's okay, you can go ahead.
Are you sure? Maybe like 10 minutes is okay?
Yeah, yeah, it's fine.
Okay.
Yeah.
The final one is I would like to share with you some kind of unexpected finding, but which is I thought is very cute and also interesting story, which is related to sugar. We know that sugar is absolutely necessary for our body. Without sugar, we cannot survive for sure, but too much sugar can be bad, sometimes even ugly. For example, they can increase the risk for many diseases such as diabetes, heart disease, so on and so forth. And we also know that sugar glucose is important for immune cell metabolism. I'm sure you heard the story by Humber-Chi in this same immune Zoom a few weeks ago. He did a very nice work regarding the metabolism of immune cells. And a few years ago, which Rick Derrick, who was one time my good friend Feng Xinhua's supervisor before, he's the TGF-beta guy. He published this paper. Actually, I met him in Keystone. I listened to his talk. I thought it's very interesting. He showed that actually the TGF-beta can be activated by this high level of glucose. Therefore, can induce cell hypertrophy. Of course, that's related to tumor. In this paper, they showed that high glucose can activate the latent form of TGF-beta through MMP pathway. Now, when I listened to that talk, I had some idea there because we know, I thought the immediate thought for diabetes, we know that diabetes is at least in the latest stage of diabetes, we have a lot of the complications in this patient. I'm sure for the people who studied diabetes know that one of the important, very knowing complication is the food ulcer. This patient is very easy to get infection. This ulcer is very difficult to treat. And then I somehow had this idea. So if high glucose can cause TGF-beta activation, could this high level of glucose maybe cause the immune suppression by induction of more t-regs through TGF-beta? That's the original idea. To test the idea, Danley Zhang, Dunfang Zhang, who also back, who very talented postdoc too, asked him to do this simple experiment by inducing T-regs in vitro by simple TCR stimulation of T cells with the medium long. This medium long means the low without adding any glucose there. Okay, we know the normal glucose, normal media, complex media, we use this with 25 millimolar glucose. That's the here. The hypothesis is whether this glucose can induce more T-regs there by inducing more for TGF-beta, active TGF-beta there. However, when he took the data as a control, he also included several other sugar like fructose and mannose. But he came back said this glucose did not induce any T-regs there, which is true. He repeated many experiments. However, what happened to glucose, we do not know. Later on, he published another paper. We know that high glucose indeed can activate TGF-beta by induce not T-regs by Th17, and being important for EAE and IBD. I'm not going to through the details. If you're interested, you can look at the paper there. However, unexpectedly, he said this Mannose can induce consistent increase of T-regs there, not only the percentage total number by 3-day culture. Now, what happened is Mannose. What is the Mannose? I had no idea. I had to go back to Google check to search what is really d-mannose. Maybe I learned that d-mannose in my biochemistry class when I was in the medical school, but I forgot totally. But the Mannose actually is quite interesting sugar because it's actually the epitomer of the glucose. Normally, it's important for a lot of things in the body. Normally in the blood, this is the 1 of 50 of the concentration of the glucose in the blood. And important for human metabolism. I'm sure a lot of people heard this Mannose receptor. For human patients, if you look at the literature by, I think this is back a few years ago, only 2 things were reported by this Mannose. One is if some patients have that inherent Mannose deficiency, you can use the Mannose as supplementary treatment for this patient. Secondly is that Mannose can be very effective to prevent or even treat the recurrent urinary tract infection, especially for women. The mechanism proposed then was that Mannose can somehow block the adherence of bacteria into these epithelial cells in the urinary tract system there. That's the mechanism they proposed. However, nothing was known about the Mannose in the immune cells, especially T cells. Now, if we see that the Mannose can induce T-reg, we have to figure out what's going on here. Since we know that TGF-beta is important for the T-reg generation, of course, we come to TGF-beta. This is one of the typical experiment which I show you here. If we get the medium long with Mannose, you can see here not very high, but consistently increase the T-reg generation. If we add the latent form TGF-beta, not the active TGF-beta, okay? Latent form TGF-beta, now itself without Mannose, you can get some generation. You can imagine that latent form TGF in the culture may have some activity TGF-beta there, but if it's Mannose, this can be increased dramatically. Now, this increase mediated by TGF-beta was, in fact, is indeed because of TGF-beta signaling because if we use the TGF-beta receptor 2, knockout T cells, all the effect was completely gone. Okay, this is not only in vitro, but also in vivo. For example, we took this adopted transport system, we inject this KJ126 transgenic T cells, which we know this over specific, into this biopsy mice. First, we feed these mice 20% of mannose in water, let them drink some sugar water, and then wait for 10 days, and then inject the T cells, and then gouge this specific over peptide for this few days, and harvest the cells in their gut. And to test whether this is TGF-beta dependent, we can induce one group with the anti-TGF-beta antibody. See what happened. As you can see here, very nice in the peripheral mesenteric lymph nodes. These are two places we can find t-regs. Most likely, this Mannose can induce very dramatic increase of T-reg generation, these cells. And if we neutralize TGF-beta, this is all gone. This is the same thing happened to mesenteric lymph nodes too, suggesting TGF-beta not only in vivo and in vitro and also in vivo play a critical role. And then the question is how that happened, how that Mannose affect the TGF-beta signaling to induce more FOX3 and induce more T-regs. We know that TGF-beta is very unique cytokine. TGF-beta mRNA increase does not necessarily mean TGF-beta signaling is increased. TGF-beta can secrete normally, majority of them is latent from TGF-beta. They have through many physiological, biological process and to become active TGF-beta. And then TGF-beta can bind their receptors to induce whenever the signal pathway. Now, which pathway around this circle does Mannose affect? First, we checked, does Mannose affect the TGF-beta increase? The answer was no. We took the naive T cells, used the control of Mannose. We did not find the increase of TGF-1 mRNA. The second is, can Mannose affect the TGF-beta receptor 1 or receptor 2, for example, increase their expression, therefore increase the sensitivity for TBA signaling? For that question, we did the following experiment. We took these over ex vivo 20, which contain no serum there, and either without Mannose, and with the different doses of the active TGF-beta. The hypothesis was, if Mannose increase their receptor expression, this TGF-beta may have a different effect. However, if you look at this, both, there's no difference at all, totally exactly same. That means Mannose at least not through this so-called increase of their receptor or increase their sensitivity. The third possibility is, can Mannose active latent form TGF-beta become TGF-beta? The answer was yes. For the same experiment we did, use the ex vivo 20 with the Mannose and add the latent form TGF-beta, but not the active TGF-beta. You can see here, without Mannose, you get some induction for FOXP2 cells, but with Mannose, this number can be significantly increased. Therefore, we believe that's the mechanism. And the next question is how that happens. We actually showed this through two pathways. D-Mannose can active latent form TGF-beta through two pathways. One is through this so-called integrin alpha-5 beta 8. Second is by increasing ROS. So this is the alpha-5 beta expression. This is ROS expression. This ROS expression is not through the glycolysis because D-Mannose can inhibit the glycolysis, but through the fatty acid oxidation. Because for time, I'm not going into details, but we also showed use knockout mice that was indeed the case. These two together actually play a major role in this Mannose-mediated generation of T-regs. Now, finally, the question is, now you play this around in vitro, but this is only for fun. Really, this Mannose can have any effect in vivo, especially for certain diseases. For that purpose, we have to go in vivo. We use the quite different model. I show you one is type 1 diabetes. We know this NOD mice can spontaneously develop diabetes. From 10 weeks or so, we started to treat these mice with 20% of D- water at 9 weeks of age. This age, although their blood sugar is not reached the diabetes level, but these mice already have massive inflammation in their pancreas. We keep feeding these mice with the water. By 23 weeks, as you can see here, the control mice, almost all of them developed diabetes. The Mannose, however, protect most of the mice from diabetes development. And this protection is indeed because of the t-regs. We use a quite different approach to prove that's the case. First one, we use the anti-25 antibody, the treatment to deplete t-regs, although this is not perfect, but we use another t-reg specific depletion approach. We got similar results. As you can see here, when you deplete t-regs, this protection was completely abolished, suggesting t-regs play a key role. We also show that's because of TGF-beta, not only for the protection, we also show Mannose somehow can also stabilize the blood sugar when the mice already developed diabetes. As you can see here, the Mannose can not let this diabetes back to normal level of blood sugar, but they can stabilize their blood sugar very well. In control, though, we can see this blood sugar can reach the high, very high RUC level. And then when we look at the pancreas, you can see here, T-regs indeed was increased, and in fact, the interferon producing CD4 or CD cells was decreased there, suggesting this is indeed important. And we also used another model, which is the adopted transfer model for the asthma. We got a similar result, which D- can also suppress Th2 type response and protect or treat this allergy and asthma in the body of the mice. Therefore, this is my final slides. I would like to summarize what I showed you here. We showed that D- can through their, actually this is the glucose transporter, also the same transporter, go inside of the CD4 T cells. And then this Mannose cannot enhance the glucose, in contrast can decrease the glucose, but can enhance that fatty acid oxidation through this pathway, can induce increase more ROS production. And in the meantime, D- can also directly to upregulate this integrin alpha-5 beta-8 expression. These two together can make more of the latent form TGF-beta become active TGF-beta. This in turn to through their receptors to induce phospholipid. This process is absolutely required TGF-beta. However, in addition to that, Mannose may also directly inhibit interferon gamma alpha production. That process is TGF-beta independent, at least not totally dependent. We still do not know what happened there. This is something we still work on that. This can somehow in vivo can do something good for certain autoimmune diseases, for example, type 1 diabetes, asthma, allergy. But I have to mention, D- is not the universal treatment for all autoimmune diseases. Some autoimmune diseases are not working. For example, EAE, IBD may not work very well. This is the commentary by Eric Peace wrote about that one. Okay, this is my last slide. This I already mentioned most of people work on that different project. If I missed anybody, I can you can find the papers yourself. This is a current member in my group there. Actually, we although we are still in the pandemic, but my group is really the international group. We come from all over the world. Somebody from France, Switzerland. This is from Pakistan, Colombia, South Korea, China, Japan, China, and the US. And I have used this opportunity, to do myself a favor. I have currently have two positions available. One is we want to hire somebody to work on T-reg or CAR-T for the tumor autoimmune therapy. Another one is try to continue our work and the immune regulation for the sensory neuron function, especially itch or pain in certain cancers, especially pain and autoimmune disease. Thank you very much. I took a little bit too long. I stop here to be happy to take any questions you have.
Okay, so thank you so much for the wonderful talk, and there's a lot of information there. I think we already have one questions. Just go ahead, Cong Lei.
Yeah, hi, Dr. Chen. Yeah, this is
Yeah.
Cong Lei from the Chinese University of Hong Kong at Shenzhen. Yeah, very great talk. Yeah, thank you for sharing with us your past achievements in this t-reg and TGF-beta field. So first, I would acknowledge that I'm not a T-cell person. Just I remember back in 2008, I read some papers regarding the controversial role of TGF-beta in the human versus mouse, the T-reg. We know that in the mouse T-reg, like TGF-beta is absolutely required. But in the human T-reg, I think that there were some controversy whether TGF-beta is important for the human T-reg induction or their suppressive function. I don't know whether this controversy already been resolved in the past. And also another question is about your model. Your model showing that the apoptotic that some sites can be engulfed by the phagocytes, then to release the TGF-beta. So I'm kind of wondering whether anyone have used a P to knock out the TGF-beta only in the phagocyte to see whether indeed phagocyte is the source for the TGF-beta, the t-reg development, and also what kind of cells are contribute to cleave the latent TGF-beta to the active TGF-beta in the thymus.
That's a good questions. Actually, I answer your second question first. I think this is something we need to. We are still working on that. Indeed, because we are now try to produce, producing that TGF specific knockout mice in the macrophages that we're doing. We have the receptor, but not the TGF-beta. Indeed, this is a very important point you made. This is the question we know we needed to resolve that. Now, I mentioned to you that we showed in the paper where the TGF-beta can be produced not only macrophages, but also dendritic cells in the thymus and even epithelial cells, which is not a total surprise. But I have to, because I have to mention, I did not mention in my talk, we know that in the thymus, depending on gender strain, the macrophages in the context and the medulla are different. I showed you that active TGF-beta staining, many of the active TGF-beta is in the medulla areas, not the cortex area. Now, people immediately can ask, we know that in the cortex, there's also apoptotic cells. And theoretically, these macrophages should also pick up and digest some apoptotic cells in the cortex. Why there's no much the active TGF-beta as in the medulla area? This is a question we have to address that too. We think we do not know what's the reason. We think this different type of macrophage may have a different role, in terms of the activation TGF-beta. Regarding how the TGF-beta be activated, now we have not had very good idea at the moment in the thymic environment, because this is definitely an important question we needed to address. Currently, we are trying to use a different approach, see what's going on. We, not only the integrin, maybe other things there, because the activation of TGF-beta from latent TGF-beta is one of the most exciting or most important area people have less studied yet. I mean, that's the important question. For your first question, the controversial argument or the role of TGF-beta between mice and human. Yes, you're right. And when people, there's no question in the mice, and you can see the TGF-beta is absolutely required. For human, there's indeed some kind of different literature. But we and Ethan Shiva's group, we both published papers. We think that TGF-beta also involved The only thing that is, the question now is that in the human T cells, so-called naive T cells, it is very hard for anybody to get the so-called real true naive T cells from human like mice, because mice you can easily get true so-called true naive T cells. When the cells are not very much of the naive, especially when they activated though, we know that from different studies, we know their TGF receptor can be decreased, changed, which may affect their effect. If you ask my opinion, we published one paper actually, we believe that TGF-beta also involved, because if you look at the literature, most of people, study that induction of FOX3 in the human T cells, they normally put this culture there and they use the complement media and put a TCR stimulation by R2, and that's it. But that they did not add any extra TGF-beta there. But can this system not be involved in terms of TGF-beta? That's maybe not, because the culture system contain TGF-beta there. For example, FBS contain large amount of TGF-beta. You have to eliminate that possibility. If you neutralize TGF-beta in that process, you may get some kind of effect. Although we have to say that human is human. It's not like exactly mice. That we have to acknowledge that it's a difference between human and mice. But my personal feeling is that indeed human T cells also need TGF-beta.
Yeah, thank you very much. Yeah.
Okay, so next question, I think from Xiaoqing. You want to go ahead and ask question yourself?
Yes.
Okay. Go ahead.
Okay. Hello, Dr. Chen. Very happy to listen the talk. I'm not a T cell aerial postdoc, so I want to ask three simple question. Maybe it's wrong. The first question is you said the t-reg and the p-reg they have a different characteristic by the different environment. So do you think there is any possibility to transfer the t-reg cells to isolate the t-reg, then transfer to the our recycle system, the blood system, then check whether they will change the characteristics to the just similar to the p-reg. This is the first question. And the second question, you already said the human in the human the TGF-beta maybe also play a important role regulate the t-reg. So we know in the Asian people the thymus size will become small and even gone. So do you think the concentration of TGF-beta will change in the Asian people? This is the second question. And the third question is because recently I read some paper about T cells, so I see that the glycolysis play a very important role in the regulate memory T cell forming. So do you think when you use can we use the Mannose to modulate the memory T cell quantity and the function? Thank you.
Can you repeat the third question? I did not quite get what is the Mannose function for?
The glycolysis have an important role in the T memory cell formation. So yeah. Can we use this Mannose to regulate the memory T cell quantity and the function?
I see, memory T cells. Okay, let's start with the third question. This is all important questions, okay? And
thank you.
That's the important question to be sure. There's never question, thinking that the question is not important, that all the questions are important. Now, the third question, I have no idea whether Mannose can affect the memory cell number or not. If you're interested, you're welcome to address. But I can see that though, Mannose indeed can compete with the glucose to affect the glucosis. For example, if we add the glucose together with d-mannose, d-mannose can inhibit the glucose induced by the glucose. Okay, that's what I can tell you. But also, there's quite different papers published already. Actually, after we published that for 2017, 2018, if people notice that in Nature, another group from UK published one paper said that d-mannose actually can inhibit the tumor growth by affecting their tumor glucosis. And also another paper a few years, maybe 2 or 3 years ago, so the d-mannose can affect the macrophage glucosis function too. So that's the third question. The second question is whether the aged people have kind of any changes in TGF-beta. Actually, this is an important question. We do not know. We do not know. We know, but we do not know. This is an important question. We do not have a chance to look at anybody have interest. We would be happy to to help or collaborate with you if anybody, especially to look at particularly whether in the human, both young and aged people, there's any changes in the serum blood in terms of TGF-beta, which I think personally is an important one, because we know, for example, the aged people supposed to have more of the R6 in the circulation, but nobody knows exactly what happened to TGF-beta. Although to measure TGF-beta by use the plasma, not serum, is very important point you have to consider. And also, I want to mention that you also mentioned that the aged people, they do not have much thymus anymore. They do have a t-reg there though. And now this is the question we still do not know where these t-regs most likely is from the proliferation and maintenance from the previous general t-regs. However, I want to mention one important point about TGF-beta. Okay, this is I use the word from Marcus Bohm, who discovered TGF-beta. TGF-beta is a switch. When something is off, TGF-beta helps turn on. When something is turned on, TGF-beta helps turn off. In terms of t-reg or FOX3 positive t-regs, if this cell does not express FOX3, TGF-beta is required for induced FOX3. But once these t-regs are generated, TGF-beta actually can suppress their proliferation. This is a very important point we have to keep in mind. That's the double soul of TGF-beta function that always happens, no matter T cells or immune cells or other cells. For the first question, it is an important question, but put this way, in the normal situation, okay, for example, spleen, lymph nodes, both in the human and mice, we know majority of the T-regs are coming from the thymus. The periphery T cells can be generated or induced to express FOX3 to become P-Tregs, but it's not always, in the peripheries, for example, the T-Tregs and P-Tregs is the distinction is that where they are produced, T-Tregs or natural T-regs, we believe these T-regs are generated in the thymus. The P-Tregs or I-Tregs are generated in the periphery, but in this normal situation like spleen, lymph nodes, maybe a lot of the tissues, a lot of the T-regs are from the thymus. That's what my answer is for that. Therefore, you said you take some T-regs, become P-Tregs. Yeah, if you take the T-regs, for example, inject these mice, you can call them P-Tregs, but these T-regs are generated from thymus. We would prefer to still think this is T-regs.
So do you think? So you think they are different when you transfer them? They can be same. You think?
No, they definitely, even for T-regs, transfer to P-Tregs, because T-regs can change their phenotype, their function, really depending where they are located. With same T-regs, for example, you inject the same T-reg into different mice. This mice is a normal mice, that mice with the autoimmune disease, that mice with the tumor, and this when you look at the afterwards, for example, the same mice in the knife mice, for example, you look at the T-regs injected from the spleen, you look at the spleen, lymph nodes, or liver, or lung, and you isolate the injected cells, their phenotype has been changed because the environment can change their phenotype. A function too, actually, yeah.
Okay, great. Thank you. Thank you so much.
Okay, so well, I think I have two questions. One is I would like to to know the comments from you because you mentioned during the T-reg cell development that the TGF-beta level kinetics in the thymus actually change, particularly increase on day 3 and start to decrease on day 4. I remember, if I remember correctly.
Not exactly decrease, but at least not very much increase. At least the day 4 we did not see further increase. I do not think that's difference between day 3 and day 4.
So you think on the start from day 3, the TGF-beta level in the thymus are maintained on the stable level?
We do not know. We only detected day 4. We did not look at the much more adult mice. We did, but I do not think that's much difference if I remember correctly. I have to go back to check.
I see. Okay, because I think it's it would be curious that if there's a dynamic or fluctuation of TGF-beta level in the thymus, how that influence the T-reg cell functionality and development. The other question actually related to the sugar story. I mean, you show that the Mannose actually can decrease the glycolysis in the T cells. I think I have 2 points related to this. One is how about the other immune cells, which also will have the same consequence. And second is, do you think this is actually specific for the Mannose or it's actually related to the other sugar, for example, the fructose?
Yeah. So second question first. The answer is Mannose may do, in terms of glycolysis, may do same thing on the other T cells too. Not only the T cells. We tested so far CD8 T cells. We have started to look at the macrophages. I know somebody already published some paper on macrophages. The Mannose can definitely affect their glycolysis similar like the in T cell CD4 T cells. And yes, Mannose is unique. It's not like the fructose and also the glucose. Glucose in terms of at least for T cell function. Fructose is more like the glucose, but the Mannose somehow is different. That's all I can tell you.
Is there any why the specificity? Is there any?
I don't know. I don't think we know that much because this is a surprise too. Actually, maybe because of their structure. Because if you look at the structure, this is different aptamer of the glucose. They have same molecule weight there. The only thing is structure is different. And why this is different? I mean, we do not have. For example, I only showed you today is the one part of the Mannose function, which is through this pathway to active TGF-beta. But you can imagine that Mannose can do many other things too. For example, yeah, that's for example, can Mannose, free Mannose bind their Mannose receptor? I don't know. Maybe this is something I can only see in that setting. We did that in vitro experiment. For example, we knew that the Mannose indeed can through the different pathway to active TGF-beta. But Mannose, as I said, can directly suppress in the gamma R4 in CD4 T cells. But that one is TGF-beta independent. Yeah, that's something else we do not know. For the first question, TGF-beta level. Can you repeat that?
No, I think you already covered the first question. My question first is the TGF-beta level fluctuation in the thymus.
Yeah, of course. And in the thymus, though, we look at it only in neonatal age. And adults, you can imagine that. I do not think that everything is very stable there. That's not possible. Depending on the environment, there must be something, because if the mice somehow get more light, so they have less sleep,
Right.
If they get more stress, get more apoptotic cells there, they may change there too. I mean, yeah.
But that way, if their TGF-beta get influence, then do you think it will also affect the Treg cell generation and the function?
Yeah. Okay. This is an important question. Now, the level of TGF-beta, especially the total TGF-beta or lentiform TGF-beta, only tell us this production of TGF-beta, cannot tell us whether that's really the changes of TGF signaling. If we think of TGF signaling in thymus, though, we have to think not only the amount of TGF-beta, the important part is we have to think of the level of the active TGF-beta.
Sure.
We also needed to think of the TGF receptor levels. A third one is that we have to think of the whether other cytokines is present or not. For example, do you have a R4, R6 around there, or interferon gamma around there? They may affect the TGF signaling too.
So based on what you said, you believe that even their environment change, their intrinsic cytokine balance to maintain the homeostasis of the Treg cells, or do you think if the environment change, the difference or the change of the cytokine can eventually impact the Treg in some way?
I think the second point is more likely.
I see.
Yeah, because whether Treg can, because we know that, for example, R2 may be the most important the cytokine to maintain the Treg survival.
Mm-hmm.
Maybe also important for their proliferation. Without R2, I don't think possible. Sure. Now, other cytokines, for example, we know that R6 can affect better than I do in terms of with R6 in TGF-beta, no matter how much TGF-beta. In the same TGF-beta, you can just shut down that factor 3 become tier 17.
Right.
4 can make tier 9. Of course, that's the beta determining factor can be influenced by other cytokines for sure.
But I think what we are discussing now is restricted in the thymus environment instead of in the entire peripheral.
Yeah, in the thymus though, it may be relatively purer than periphery. Yeah, because R2 is there. We know that R2 is there. Mm-hmm. How much are R4, R6, or other interferon gamma? I wouldn't think that too much. Maybe R17 has something there, but R17 so far we do not have any evidence R17 can affect the Treg or factor 3 expression.
Sure. Okay. I think another question from Hui Yuan. You want to just ask question? I see you raise your hand. Okay. Well, if you don't have questions, then
just give me one. Yes, just give me 1 second. I just unmute my microphone. Ahead. Hi, Wanjun. This is Hui Yuan from Chengdu. Long time no see. I only have one short question regarding Mannose. So have you ever checked is Mannose have direct effect on macrophages to stimulate either TGF production or maybe relate to secrete more MPP and somehow active latent TGF-beta?
Yeah, that's a very good question. I think I have a fellow who are starting to look at that question, although I posed him for a long time. We're starting to look at that question. We have not got any data yet. But I remember, if I remember correctly, there's last year or the year before, some publication of some people already published one paper by seeing that Mannose can affect the macrophage function. I did not read the paper very carefully. You might want to look at that paper, see what they have done so far. But my personal feeling is that there's no reason that Mannose does not affect the macrophage function, even for activity TGF-beta 2, because we know the macrophage is one of the most important immune cells producing TGF-beta, much more than T cells.
Okay, thank you very much, and hope to meet you soon in China. We all miss you, really.
I hope so. Thank you.
Yeah, thank you.
Okay, well, I think that will be.
Let's on chat. Somebody or has question now?
Okay. I think that's all for today. So I want to thank you so much for the seminar. I think we learned a lot, and then I think a lot of friends can catch up. I really appreciate it. So Thanks, Wanjun, for the wonderful seminar. And then I hope see you all next time.
Thank you. Thank you.
Yeah, thank you.
Thank
Yeah.
you, everybody.
So have a good evening.
Thank you. Bye-bye.
Bye.
Keep in touch. Be safe, though.